No.
Hi there, hi everyone. Good evening. Most of you have joined now. I guess we can start by now. Today, as we discussed yesterday, we're going to discuss about chip analysis and southern blotting. So these two topics will be covered in this topic, and later on we can discuss about facts analysis in the evening part at the 9 p.m. I hope you have good weather. Here we have a lot of sunny, bit hot, humid, without rain as usual, right? So chromatin immunoprecipitation chip analysis. So what is chromatin immunoprecipitation? That is chip. It's basically done to check the your DNA and protein together, how much they are interacting with each other. They're interacting by transcription factors. They're interacting by others factors. So in that case, we do this chromatin immunoprecipitation. So you have your cells, then you cross-link with your DNA, right? And then you lyse it with the lysis. Then you add your towards your DNA, you add your um, yeah, cross linkers, and then with the antibody you join with these cross linkers with the antibody. Then you immunoprecipitate these linkers, then you wash them, and then you have cross linkers reversible. Then at the end you have to DNA purify and then quantitate with PCR, and then at the end you can check this DNA and protein analysis with this technique. So not only protein interacts with one another, they can also interact with DNA. So in that case, to see the interaction of a protein with the DNA, chromatin immunoprecipitation is a technique that determines whether a protein of interest interacts with a specific DNA sequence or not. So this technique is often used to study the repertoire of sites on DNA that are bound by particular transcription factors or by histone proteins, and to look at the precise genomic equations of various histones and modifications. So how does CHIP works? So CHIP can be used to examine the presence of protein DNA interaction at steady state, or to quantify changes in interaction at specific phases of cell cycle or following treatment of interest. So protein and associated chromatins are temporarily cross-linked in the cells and the living cells or the tissues using formaldehyde or UV light, and then they sheared using the enzymatic digestions or sonication to yield around 300 to 1000 base pair fragment of DNA. Then protein of interest along with any associated DNA fragment is immunoprecipitate from the cell debris using the specific antibody. Then the cross-link is then reversed and DNA fragments are purified, and the amount of eluted DNA can be assessed through the quantitative real-time PCR using primers flanking the genomic locus of interest. Then DNA amplification is an indication of enrichment and binding of the proteins of interest. Then modified this chip technique could be modified according to the things that you're looking for. These protocols are modified a bit. So first of all is chip on chip, which is a genome wide analysis of protein binding sites using micro analysis of purified DNA fragments. Then chip sequence, genome wide analysis of protein binding sites using deep sequencing of purified DNA fragments. The native chip, which omits the cross-linking step and uses micrococal nucleus digestion to cut DNA at histone linkers to examine the DNA target of histone modifying protein. Then chip exo, addition of an exonuclease digestion step to obtain increased resolution of protein binding sites up to single base pairs. And there is chia PET, which is a chromatin interaction analysis by paired and end-text sequences. So a technique that combines the principle of chip with chromosome conformation capture to detect long-range chromatin interaction mediated via protein of interest. Then iChip, indexing first chromatin immunoprecipitation, a highly sensitive technique that reduces the number of cells required for chip experiment by initially barcoding total cellular chromatin. An N-chip, which is an engineered DNA binding molecule mediated chromatin immunoprecipitation, which is a technique which employs the CRISPR-Cas9 system to target specific genome region. Then RIP chip or RIP sequence, this is to see the interaction between your protein and RNA. So some limitations of this chip technique. As with the molecular biology technique, chip is not without its own limitations. Chip assays often yield low signals as compared with the controls, leading to the inconclusive data. And chip will infer the presence of proteins at a given genomic locus. It cannot be determined functional significance of protein binding at that DNA region. And cross-linking may additionally include proteins that transiently interacts with DNA or DNA binding proteins have no functional significance. And similarly, DNA interactions of proteins with short residence times may not be fully captured in them. And additionally, interacting proteins may mask the epitope of the protein of interest. Finally, the chip technique is extremely dependent on the quality and specificity of the antibody employed and may not discriminate between different DNA binding protein isoforms. Some important factors to consider. So while the principle of chip technique is quite simple, the actual execution of chip experiments can be rather complex. So here are some important factors to consider when planning or troubleshooting your chip assays. First, garbage in and garbage out. So that the quality of a chromatin generated in chip can make difference between an interpretable results and quality data point. Antibody validations, while antibodies for Western proteins can recognize your denatured protein, so chip requires recognition of the tertiary structure of protein of interest and of exposed epitope. Then cell number, that you should have particular number, optimized number of cells while doing your studies, so that histones modifications are quite abundant, and chipping for them they require less material. It could be experiment could be done nicely. There could be cross-linking issues, that they could interact with another thing instead of other, and it could lead to different pull down then. Need to use appropriate controls like IgG, input controls, positive control locus and negative control locus. So let's check this one. Something in your ear about ChipSeek. Yeah. StatQuest. Yeah. Hello, I'm Josh Starmer and welcome to StatQuest. Today we have a gentle introduction to ChipSeek. Note, this StatQuest is based on the gentle introduction to RNA-seq, so watch that first unless you're already totally down with RNA-seq. Okay, we've got a bunch of cells. Inside each cell, there's a nucleus. And inside each nucleus, there are a bunch of chromosomes. Let's focus on the chromosomes. Specifically, let's focus on these chromosomes. Chromosomes are made out of chromatin. Chromatin is made out of DNA, plus histones, a type of protein, plus other proteins that we'll talk about some other day. They're not that important for understanding ChipSeek. DNA wraps around the histones to package DNA, and the packaging can regulate gene transcription. Depending on how they are modified, histones can activate or repress genes. Lots more can be said about chromatin and how it's packaged, but that's for another day. Today we just need to know that chromatin is essentially DNA wrapped around histones. To simplify things, let's just use this big blue line to represent chromatin from here on out, but remembering that it's made of DNA and histone proteins. And let these big brown arrows represent genes. And let this green circle represent a histone that allows transcription. And let this red stop sign represent a histone that represses transcription. In a cell, all kinds of proteins can bind to DNA. This mustard-colored thing might promote gene transcription of this gene. And this pinkish-colored thing might repress this gene. And who knows what this green thing is doing? We can use ChipSeek to find out. ChipSeek stands for chromatin immunoprecipitation combined with high-throughput sequencing. It identifies the locations in the genome bound by proteins. That's the most important thing in this StatQuest, so I'll say it again, in bold. ChipSeek identifies the locations in the genome bound by proteins. For example, say like we wanted to identify all the regions in the genome bound by the green thing. The first thing we do is use formaldehyde, or something like it, to glue all the proteins bound to the DNA together with the DNA. This means that all of the DNA-bound proteins, including the ones we're not interested in, are glued to the DNA. The next thing we do is cut the DNA up into small, approximately 300 base pair, fragments. Then we isolate the protein we're interested in this case, we're interested in the green thing, using an antibody. The black star represents an antibody that is attached to a bead. Then we isolate the proteins bound by the antibody and wash everything else away. Then we reverse the formaldehyde glue by warming up everything. Then we isolate the DNA by washing away the proteins, including the histones. Now that we see how to isolate DNA that is bound by a particular protein, let's take a step back.
So far, the example has focused on just these chromosomes. But the process, glue proteins to DNA, cut up DNA, bind proteins of interest with antibodies, isolate antibodies, unglue and wash away proteins, applies to all the chromosomes in the cell. And it is usually applied to a pool of 6 million cells, give or take a few. So we end up with a lot of DNA fragments from a lot of cells. Then, just like with RNA-seq, we prepare a sequencing library by adding sequencing adapters to both ends of the DNA fragments. Then, just like RNA-seq, we PCR amplify the library, check the library concentration, sequence, filter out garbage reads, and then align the high-quality reads to a genome. That is to say, if this is the genome, the first read might come from here, a location on chromosome 2. The second read might come from here, a location on chromosome 1. The third read might come from here, another position on chromosome 1, etc., etc., etc. Ultimately, we get a long list of genomic coordinates for all the reads, usually between 50 and 100 million reads. And we can use those reads to create a genome browser track. These are genes and chromosome positions in the mouse genome, MM10 to be exact. This is the track that we created for our ChIP-seq reads. A lot of reads map to this region, and relatively few reads map to these other regions. This track was made from a control experiment. The control track was made by taking some of the input chromatin from the original ChIP-seq experiment, and, without using an antibody to enrich for any particular protein, ungluing all the proteins and washing them off. Then sequencing, aligning, and converting into a track. In summary, the control track uses some of the same input chromatin for the ChIP-seq experiment, but doesn't try to enrich for any particular protein binding. We use the control track to verify that a high concentration of reads in the ChIP-seq track is due to a protein binding there, and not because a lot of reads map to a repetitive region. Statistically significant peaks are usually represented on genome tracks by rectangular bars. We could then compare peaks for the same protein in different cell types, like lung versus kidney. Or, if we didn't know the specific DNA sequence that this green thing bound to, we could guess that it is a motif found in all of the peaks. Here's a motif found within the peaks that indicate where the green thing bound. The large letters are more frequently associated with the green thing than the little letters. We can also try to determine the functional role of the green thing by looking at where it binds relative to the genes. Here, we see that the green thing binds near the start of a gene, so it might regulate that gene in some way. Anyway, those are some of the things you can do with ChIP-seq. In summary, ChIP-seq identifies the locations in the genome bound by proteins. Bam! Hooray! We made it to the end of another exciting stat quest. If you like this stat quest and want to see more, please subscribe. And if you want to support StatQuest, please click the like button and consider buying one of my original songs. Okay, until next time, quest on.
Okay, I hope this chip sequence is clear now. Might be doing it in future, in your future lab, or in internship. This is quite a future in its upcoming future. Now let's switch to blotting technique. So just remember that we have discussed also before snowdrop, S for southern, N for northern, W for western, D for DNA, R for RNA, P for protein. So in this way you can remember that which technique belongs to which one. So these techniques, as we discussed, they are done to detect DNA, RNA, and protein, and specifically designed for the same. So we will discuss how we do these techniques in particular and what are the various ways to do so. So we will discuss in general the PPT and then one virtual video also about that. Yeah. So southern blot hybridization detects target DNA fragments that have been size functioned by the gel electrophorese. So this technique was invented in 1975 by E.M. Southern. So principle is in this technique, we exploit the property of radio level probe with a single stranded DNA. If we want to detect the presence of simple sequence in our mixed DNA sample, then we will accordingly design the probe which will have complementary sequence to our target sequence. The procedure is that in this procedure called southern blot, DNA from sample is cleaved into restriction fragments with a restriction endonuclease, and then fragments are spread apart by the gel electrophoresis. So here is the principle. So first you run the gel, then you will see your nucleic acids on your gel by the electrophoresis, then you transfer over the nitrocellulose membrane. Then after transferring, you put over the radio level nucleic acids, and then you can detect them with the help of X-ray rays, RT radiography. So the information that we get from the southern blotting is very essential. We can get to know about which genes are present, how many copies of these genes are present in an organism, the degree of similarity between chromosomal gene and probe sequence, whether rearrangements have occurred during the cloning process, and so on. Further, it helps to detect the certain cancers and genetic diseases such as monoclonal leukemia, sickle cell mutation. Beyond that, used in the DNA fingerprinting, genetic engineering, and forensic science for tests as under paternity testing, personal identification, sex determination, species exclusion. So let's check how does it done. It will not have any sound. First, you do your GNA, DNA, gel electrophoresis with your samples. Okay. So now next is your that is your RNA, that is northern blotting. Is the same technique, is just we use little bit of different um, level of things in that. Does it hang or what? So the northern blotting is a simple extension of southern blotting, and it derives the name from the earlier technique. It's one of the technique in molecular biology in which principal aim being the measurement of your RNA. So RNA molecules are separated by size and detected on the membrane using a hybridization probe with a base sequence. So RNA is first extracted from the cells of interest. Then precautions has been taken care to avoid degradation of single-stranded RNA by ribonuclease, that is RNase, which is then founded on the skin and glassware. Then you need to wear gloves, use specially treated plastic and glassware to avoid accidentally introducing ribonuclease to extraction prep. Then you additionally add diethyl pyrocarbonate (DEPC), which inhibits ribonuclease activity and also bakes at high temperature, which destroys ribonuclease activity. So the same steps, same things, everything is same. I will play the things related to that.
I'm on my knees when I'm begging.
Yeah. So it was almost the similar technique like your DNA, southern blotting. Now let's check the other technique, that is your western blotting. It's going a bit slow. That's the last technique. So Western blotting is the technique which is done to analyze specific proteins that are present in a given sample, and we can interact with that. We can run these samples under denaturing and native state also. So basically this was discovered by the George Stark laboratory at Stanford and given by the W. Neil Brunett, and the name is plain name is Southern blotting, and then Edwin Southern because of that. So it is an analytical method where protein sample is electrophorized on SDS page and electro transfer onto the nitrocellulose membrane. Then transfer protein is detected using specific primary antibody and secondary enzyme labeled antibody and substrate. In procedure, first of all, sample protein is separated on the basis of the molecular mass using STS page or two-dimensional electrophorus. Electrophorus moves the protein from the gel onto the nitrocellulose where the proteins adhere. Then to detect the specific protein, an antibody to that protein must be available, and the nitrocellulose membrane itself has non-specific sites that can bind proteins including antibodies, which must be blocked with a non-specific protein isolation. The primary antibody is added into the milk solution and binds to the solution protein of the interest, and the antibody protein complex is detected using secondary antibody that has label attached to it. So here we can see after transferring your proteins over the nitrocellulose membrane, you run with the sample with the primary antibody, then with the secondary antibody, then you can watch under the expose to detect the location of your secondary antibody, and hence you can see your protein in that. So it has many applications. So now let's check how the Western blotting technique is done in practically, and that will be the last video for today.
In this video, you will learn how to perform a Western blot. A Western blot can be used to identify specific proteins in a sample and provide information about the protein size and relative abundance in the sample. First, fill a tray with blotting buffer. You will be using this buffer to equilibrate your gel prior to starting the Western blot. Next, remove the gel from the gel cassette using the opening key. Line up the arrows on the opening lever with the 4 arrows on the cassette to open the cassette. After trimming the top and bottom of the gel with a straight edge, equilibrate the gel in the tray with blotting buffer for 15 minutes on a rocking platform. Pre-soak fiber pads in blotting buffer so that they are thoroughly soaked. To make a blotting sandwich, obtain a container large enough to fit the gel holder and add enough blotting buffer until the container is filled approximately 1 centimeter deep. Place the gel holder cassette in the container with the black side down and immersed in the buffer, and the white side up and out of the buffer. Lay 1 fiber pad flat on the black plastic. Next, wet a piece of blotting paper and place it on top of the pad. Be careful to avoid any bubbles between the pad and the paper, and make certain the buffer covers the paper. Take the gel and carefully place it squarely onto the blotting paper. Again, being careful to avoid any bubbles between the gel and the blotting paper. Next, you will be applying a piece of nitrocellulose membrane. Remove the protective sheet from the membrane and wet the membrane with blotting buffer. Carefully place the membrane squarely on the gel. Avoid moving the membrane once placed on the gel, as proteins will begin to blot immediately. Using a roller, remove any air bubbles between the gel and the membrane. Place a second sheet of wet blotting paper on top of the nitrocellulose membrane. Place a second wet fiber pad on top of the blotting paper. Fold the clear plastic side of the gel holder over the sandwich and clamp it to the black plastic side by sliding over the white clip. This tight fit will squeeze the sandwich together. Insert the gel holder into the inner module. Make certain that the black side of the gel holder is next to the black side of the module. Place the inner module into the electrophoresis chamber. Add a frozen cooling unit and fill the chamber with blotting buffer to the level of the white clip on the gel holder. Place the lid on the electrophoresis chamber. Connect the electrical leads to the power supply, making sure the connections are correct, red to red and black to black. Turn on the power supply and run the blot at 20 volts. If a timer is available, set it for 2.5 hours. When the run is complete, turn off the power supply and disassemble the electrophoresis chamber and remove the inner module. Open the module and place it in a container filled with blotting buffer with the black side down. Starting with the first fiber pad, remove each layer until you reach the nitrocellulose membrane. As you remove the membrane, note that the proteins have been transferred from the gel to the membrane. Note that the kaleidoscope pre-stain standards have been transferred and can be seen on both sides of the membrane. You can also see that there are no longer any proteins on the gel. Immerse the membrane in 25 milliliters of blocking solution and incubate it for 15 minutes at room temperature on a rocking platform. Pour off the blocking solution. Add 10 milliliters of primary antibody. Incubate for 10 to 20 minutes on a rocking platform. The platform should be set at a faster setting to ensure constant coverage of the membrane. Pour off the primary antibody. Rinse the membrane quickly in 50 milliliters of wash buffer and then discard the wash buffer. Add Add another 50 milliliters of wash buffer to the membrane and let it wash for 3 minutes on the rocking platform at a medium speed setting. Discard the wash buffer. Add 10 milliliters of secondary antibody and incubate the membrane for 5 to 15 minutes on the rocking platform at a fast speed. Pour off the secondary antibody. Rinse the membrane quickly in 50 milliliters of wash buffer and discard the wash buffer. Add an additional 50 milliliters of wash buffer and wash the membrane for 3 minutes on the rocking platform on a medium speed setting. Discard the wash buffer. Add 10 milliliters of substrate. Incubate the membrane with the substrate for 10 to 30 minutes with either manual shaking or on a rocking platform. Watch for the color development. Once the colors have developed, rinse the membrane twice with distilled water and blot dry with a paper towel. Air dry for 3 to 60 minutes and then cover in plastic wrap for storage.
Yeah, so that was uh, the Western blotting. Now comes the last part, the mixture of everything. There is Southern Western blotting is done to see the interaction between your DNA and protein. The Northern Western to see the interaction between your RNA and protein. And there's a dot blotting which is done to find the it's a modified version of Western blotting technique to identify the protein of interest, and the dot methodology is done to find out the traditional Western blot techniques by not separating protein samples using electrophorese. Then comes your zoo blotting. So zoo blotting is based on the principle of Southern blotting. So in the genome of any organism there are two regions, the coding and non-coding regions. It is the coding region that causes interest for most of the researchers, as it is associated with the genetic information for specific protein. But the problem is that most of the regions of DNA are non-coding, and the question is how to identify coding regions in large amount of non-coding regions. So zoo blotting is precisely used to distinguish coding DNA from non-coding regions, and during the evolution so here we can see that there is a rat, cow and dog and human. So we both have coding and non-coding regions. But when we run these probes over the non-coding regions, so we could see mainly the humans were there. But we ran over these species for the coding DNA, we can see all these sequences coming up. So we can compare that the rat and the human, they have the almost similar coding region, and we can interpret some data out of it. So that's why it's like it says zoo blotting. It's all organisms are run to check which part belongs to which one and which is corresponding to the human part. Yeah. So that's it from today perspective. I think will not take much more class, and yeah. So in the evening, so we just today discuss your chip sequencing and your all blotting techniques. So somebody was asking initially in the first class to me that do we do sir any blotting techniques or electrophoresis techniques. So we are done with almost all. So we did so many electrophoresis technique and details also. So I hope This will be quite beneficial for you. You have learned out of them. Now we are coming to the next part, that is facts analysis. Shall we continue the class now, or you have a class of vaccine technology? If we start the class of facts analysis now, so we will not have a class at 9. Do we want to make me class now, or we do it after having some break at 9 p.m.? Because that will take hardly me 40 minutes for that class, for the facts analysis. As per recommendations, I will do it. If all suggest, so they are all almost like yeah, 30 slides in this, in facts analysis. So shall we continue, or okay, so you want to go? Actually, I can see. Okay, we see us at 9 p.m. for the facts analysis then. Yeah. So thank you. Take care. Bye bye.